So most of you know who I am and know about the program, but there are a lot of people in the audience that I don't know, and so I'm guessing that this will be your introduction to what ONR is doing. So ONR is sort of the lead organization within the Navy for funding research that supports the naval forces and future capabilities we'll provide for them, as well as for the Marine Corps. We have the ability to support projects at the fundamental research level, as well as applied and then advanced prototype development, which is really valuable as you have promising basic research to at least have the ability to go on and do some of the more translational work. So several years ago, I was at an applied environmental microbiology Gordon conference. I think it was 2010. And Janet Jansen was hosting a symposium, a session on human microbiome. And until that point, I'd managed a fair amount of environmental microbiology, but never any human-oriented microbiology. And this microbiome concept really fascinated me and got me thinking about what could ONR do in this space. And maybe a couple of years after that, I became really excited about the gut-brain axis and the role that the microbiota could play in that interchange between the gut and the brain. And so some of the studies that inspired me at the time were really looking just at animals, but understanding the role of the microbiota on central nervous system development and brain development, on behavior and mood, cognitive function, fatigue, pain relief, gender differences in some of the studies. And as time has progressed since 6 or 7 years ago when we first started thinking of this program, we've now seen more human studies sort of supporting the notion that the microbiota play a critical role in this axis. Lieutenant Colonel Hoisington mentioned sort of the unique environments that the military has to operate in, and I have a couple of examples here, just to get the academic folks thinking about this. So we look to fund research in spaces that NIH isn't going to do or NSF isn't going to do, and so we're driven by what our personnel are being exposed to and the conditions under which they have to operate. So expeditionary operations, rapid changes in environment. It could be altitude, temperature, time zone. I work for the Navy, so we think about submarines and ships. Submarines are really interesting, really different environments. I think from a built environment perspective, it would be really cool to study them. I don't know if we're going to be able to do that, but we think about people who have to work on an altered circadian rhythm, so 18-hour work days, no sunlight, very close quarters, people sharing bunks, elevated CO2, and now they also have females on submarines. And then just general combat situations, long periods of time without sleep, sporadic nutrition, perhaps operating while you're in pain, anticipating the fight to come. So these are sort of a unique set of stressors, I think, that distinguishes the military funding apparatus and sort of the community that we serve. So my program is driven by the question, does the gut microbiome play a role in the response to these types of stressors? So this is just articulating that here again. So we want to explore the effects of various stressors on the host and its commensal gut microbiome. And the impact, we hope, in the end, is to be able to increase warfighter resilience by manipulating these gut microbiomes, and we could do that in a number of ways. So, boy, it's hard to see the screen from the podium. So we are interested in characterizing the physiological, psychological, and cognitive responses of both the host and the gut microbiota to these stressors. We're looking at both psychosocial stressors and physical stressors. So the psychosocial ones include fear and anxiety and sort of crowded living conditions. Physical stressors, things like sleep disruption or deprivation or circadian rhythm disruption. We are emphasizing we want to increase our understanding of the gut microbiota brain axis signaling. How is that happening? And we see an important niche in developing tools to enable us to study the interactions in more detail. So what kind of signaling is occurring and how is it occurring between the microbes and the epithelial cells in the gut? And then also part of the program is to explore how one might manipulate the gut microbiota to enhance resilience of a person to stress. So we are thinking about different prophylactic or interventional strategies that would mitigate these effects on the host. So the technical approach is shown here. Again, I think these are very complex systems, and you have to be sort of ambitious in your thinking of how you're going to tackle them. But we are fortunate that we can look at animal models, which are every few weeks there's a new model coming to light that helps us study these stressors. So the suite of germ-free, notobiotic, humanized, genetic mutant animals helps standardized tests for measuring psychological and cognitive effects as well as physiological condition in the animals. A whole suite of methodology that you've heard about in detail from some of the previous speakers, and you'll hear from others. End goal, like many others, trying to link specific functions to the microbes that are present. We have very limited human studies, and Lieutenant Colonel Hoisington mentioned a MURI. So that acronym is Multidisciplinary University Research Initiative. So those are large team projects that they really have ambitious goals. They are, by definition, multidisciplinary. So we have a project with the University of Colorado Boulder, so Dr. Lowry is part of that effort. That particular effort does have some limited human studies looking at the effect of sleep deprivation. So for all the studies, we're interested in measuring the microbes and their metabolites. And as I said, with the human studies, fortunate they're also looking at some intervention strategies, so pre or probiotics and how they might affect the resistance, the tolerance to stress. So DoD relevance, I explained some of these unique stressors that we expect are not just affecting the host, but their microbiome. We need to understand the effects, the timing and magnitude of these effects, because they could be very important to our mission. And you'll hear more about that from some of the other speakers. We would like to think that manipulating the microbiome may allow us to enhance fitness of the warfighter under different conditions. That remains to be seen. And I'll just mention here, it's not an explicit focus of my program. I do have a few projects looking at intestinal diseases or disruptions, but diarrhea caused by gut infection and inflammation is like one of the most significant issues affecting the Department of Defense. So this program is interesting from an O&R perspective in that a lot of special programs have enabled me to fund this work at a level of several $1 million a year. So MURI is a large program funded by the Office of Secretary of Defense. We also have a basic research challenge program internally funded at O&R, and then several young investigators working in this space. And so this is just a laundry list of the types of projects that we're funding. I haven't heard the previous speakers mention synthetic biology or symbiotics. We do have a number of young investigators that are, again, in terms of pushing that tools front forward, looking at engineering microbes to serve as sensors of the gut environment, or to actually manipulate them to be sense and respond tools, if you will, that are going to monitor for presence of a pathogen, for example, and then make an antimicrobial peptide or other molecule to kill a pathogen in the gut, or developing tools to do the spatial biogeography of the microbes in the gut and how they interact with the epithelium. And so we have a lot of really talented early career scientists, and that echoes what Paula mentioned in her talk, very important. So we kind of like to take risks. So some of what we support, it may not come to pass as being an important capability for us, but I think what we're doing is adding to the general knowledge level for the field. So just some highlights from the program. There's a short list here. So we're supporting some work at McMaster University. They're looking at some specific probiotic strategies, a Lactobacillus rhamnosus strain that appeared to decrease stress-induced anxiety-like behavior in mice and prevented deficits in social interactions. They also did some work with early life exposure to clinically relevant doses of antibiotics that were very interesting in mice and show that there may be sort of a permanent effect on the gut microbiota from those exposures. So coming out of the MURI, which involves not just University of Colorado, but Northwestern and UCSD, sleep and circadian rhythm disruption altered microbial structure in the metabolome and the host metabolome in rodents and in humans. That was kind of an interesting finding. On the synthetic biology side, Jeff Tabor at Rice University engineered a gut microbe to detect markers of inflammation and could show that basically this was a marker that nobody really knew was associated with inflammation, but by engineering the microbe to detect it and then report on it, kind of confirmed that it was involved, and that was in a dextrans sulfate model of inflammation in mice. What else? So at the Pacific Northwest National Labs, a study run by Janet Jackson looking at a unique mouse model, a collaborative cross model, kind of sort of connecting together the effects of early life exposure to diet and home cage effects, host genetics and diet on the gut microbiome and the metabolome. So looking not just at the microbiome and its relation to the host, but the genetics of the host and how that influences the microbiome that populates a host, which is pretty interesting. And then also effects that involve the host genetics and the microbiome and the immune system, which I don't think they really expected to see. And then we have here in the audience Victor Chan and Cam Mousey, who've been working on a new rat model that has de-individualized humanized microbiome, and that's successful, and I think you'll hear about that from Cam either today or tomorrow. And then some work coming out of Harris Wang's lab at Columbia University directed at this sort of microbial biogeography mapping, understanding where the microbes are in relationship to each other as well as to the epithelial cells in the gut. So this is just a laundry list of the projects. I'll leave that up there for you to look at, but I don't want to forget to mention we also have so Office of Naval Research as well as the Air Force and Army scientific research offices we have locations around the world, and so we're fortunate to have Monique Baudouin from the ONR office in London. Raise your hand, Monique. So Monique, so the important thing about those global offices is they can fund research with overseas institutions. So they can fund collaborative research with an Italian group, for example, working with a US group. So she actually has some work in gut microbiome being funded in Italy. So that person is looking at a way to monitor metabolites in the brain, circulating in the brain that are gut metabolites. So injecting metabolites into the gut and then looking for whether or not they show up in the brain. So I wanted to mention that because you may have collaborators in other countries, and getting some funding to them to let them work with you could be an important mechanism for you. So I'll only be here till 11:30 tomorrow, but if you have questions about the programs, happy to answer them later today or at the dinner. Thank you.